Today you discussed about polyploidy. What is polyploidy? An organism in which the number of chromosomes set is higher than the diploid number. That is called polyploidy. That means every organism has a set of chromosome number that is 2n. Every organism has a set of chromosome that is 2n. When that 2n exceeds from 2n by 1 or 2 or loss 1 or 2, that is 2n plus 1, 2n plus 2. In that case, the chromosomes number exceeds to normal level, that is called polyploidy. That means if the number of set of chromosomes exceeds or than the normal level, then that is called polyploidy. Characters of polyploidy. In case of plants, that is, we have to talk about that increase in number of floral parts. That is, every floral parts are in five petals, 5 sepals or 4 petals. In some cases, 5 petals exceeds to 6 petals. 5 petals exceeds to 6 petals. In that case, that is a polyploidy. If flower is a normal shape is a small size, if that flower is a big size, then that is also a polyploidy. Leaves are if large, or large means that exceeds to normal size. Leaves are large, thick and deep green. Every part that is shape, the thickness, the greenness is exceeds to the normal level, then that is also a character of the polyploidy in case of plants. In that formation of a large number of pollen grains. Fruits and seeds much larger. Fruits are, if the shape of fruits or size of the fruits exceeds the normal shape and size, then that is also a polyploidy. Increase in cell size with more prominent nuclei. Increase in osmotic pressure of the cell shape. Cell shape means the liquid part of the cell, plasmatic part of the cell, that is called cell shape. And every liquid parts or cell shape exists a pressure towards to cell wall or towards to the outer membrane, that is called osmotic pressure. And if osmotic pressure exceeds to the normal level, that is also a character of polyploidy. High concentrations of the minerals, that is calcium, magnesium, potassium, these levels if increases, then the normal level, the elements, that calcium, magnesium and potassium, any other elements may that also if we exceeds to normal level, then that is called your character of polyploidy. Types of polyploids. There are two types of polyploids. We take euploids, and another is neuploids. One is here euploids are those forms in which the chromosome number has changed in such a way that an organism has an exact multiple of haploid number, such as triploids, tetraploids, pentaploids, hexaploids, and so on. These are your euploids. That means organisms in which the chromosome number has changed such that organism has an exact multiple of haploid number. multiple of a haploid number. That means triploids. It is a multiple of 3. Tetraploid. It is a multiple of 4. Pentaploid, multiple of 5. In this way, that's and so on. See this case, the organic chromosome number change in such a way that then it is called euploids. Now come to neoploids. Neoploids, that is called heteroploids, are those forms in which chromosome number has changed in such a way that an organism does not have an exact multiple of the haploid number. Is not an exact multiple of a haploid number. For example, 2n minus 1. It is not a multiple of 2, 2n minus 2, or 2n plus 1, 2n plus 2. These are not exactly multiple of or 2, 1, 3 like this. So these are called as neoploids. Euploids has been used in plant breeding and improvement of work. Euploidy has been used in plant breeding and improvements of plant biotechnology, plant breeding or plant biotechnology, improvement of work. Euploids are of two types, autopolyploids and allopolyploids. So euploids are of two types, autopolyploids and allopolyploids. And what do you mean that autopolyploids? There is an exact multiplication of one and the same genome as shown below. As shown, that is multiply one and the same genome. That is AA refers to AAAA, autotetraploid. That means 2n duplicates into 4n's. It is a autopolyploids. In allopolyploids, there is a cross between two diploids. There is a cross between two diploids, different genomes, as in interspecific hybridizations. That is capital A crossed with capital B. We have to get capital A, capital B. That is 4n. It is also called allotetraploid. Allotetraploid. Segmental allopolyploids are intermediate between auto and allopolyploids. Autopolyploids and allopolyploids is an intermediate product, that is segmental allopolyploids. So in this way, in these allopolyploids, the different genomes which are present are not quite different from one another. Chromosomes from different genomes do pairs to some extent, and multiplans are formed. Stabins are called as allopolyploids as a segmental allopolyploids. Segmental allopolyploids. Then induction of polyploidy. Polyploidy in plants can be induced by colchicine treatment. Colchicine is an alkaloid obtained from species Colchicum autumnale. It is a plant from the Liliaceae, Colchicum autumnale. Colchicine inhibits the formation of spindle in the dividing cells, and hence chromosomes do not separate at anaphase. Thus, a restriction nucleus. Is formed. That restriction nucleus is a nucleus in which chromosomes have divided but could not separate into two daughter nuclei. Effect of colchicine is temporary. As a cell recovers from the treatment, a new spindle forms and the restriction nucleus undergoes normal mitosis as a polyploidy cell. So you know the colchicum or colchicine is a chemical obtained from the colchicum automanili, that is a alkaloid, and it inhibits the process of mitosis as it inhibits the restriction of spindle formation during the anaphase. Hmm. It is a inhibitor of mitosis, or it provides checkpoints for the mitosis, so that cell division is restricted. Then you now come to characters of autopolyploids. Autopolyploids are characterized by the presence of same characters as a diploid parent, except that they are large replica of the diploid parent. They show large flowers and fruits. Cell size, stomata size, nuclei, etc. are larger than the parents. Chloroplasts are more polyploid cells than the diploid cells. Cytologically, autopolyploids are identified by the presence of multivertex. Autotriploids are highly sterile. Autotriploids are highly sterile because the random segregation of 3 chromosomes. Autotetraploid shows the sterility. Autopolyploid shows the sterility to some extent because of irregular segregation of quadrivalents by lagging of the univalents. Then you come to characters of allopolyploids. Allopolyploids show a combination of characters of 2 parental forms. There is nothing like zygnatic effect as seen in autopolyploids. F1 hybrid is completely or partially sterile. Chromosome doubling in F1 hybrid restores the fertility. Role of autopolyploids in plant improvement. Autopolyploid has seen to increase the seed size and cereals and pulses, root size in root crops, flower size in ornamental plants, seedlessness in fruits, and the quantity of active ingredients in medicinal plant. That is, autopolyploid has an use increase the size of cereals and pulses, root size in root crops, that is flower size in ornamental plants, seedlessness in fruits, and quantity of active ingredients in medicinal plants. In some fruits, seedless varieties are desired, as in grapes, guava, and watermelon. Since triploids are sterile, triploidy is used in such cases for developing seedless varieties. Seedless varieties is a character of sterility or sterile character. Then now come to role of allopolyploids in plant improvement. Allopolyploidy is important in interspecific and intergenetic hybridization. Allopolyploidy is important in interspecific and intergeneric hybridization. Allopolyploid cultivated plants include wheat, sugarcane, cotton, tobacco, and sesame. General rays through the intergeneric allopolyploidy include triticale or triticum. Triticale is cultivated from triticum and sickle, and Raphanus brassica is cultivated from Raphanus and brassica. Origin of cultivated hexapolyploidy breed wheat. So that is also a character of that. Uh, a wild and cultivated wheats from a series of diploid AA, that is 2n is equal to 14, tetraploid AABB, that is 4n is equal to 28, hexaploid AABBDD 6n minus 42 types with the basic number 7. The basic number 7. The bread wheat belongs to the hexaploid group and has an originator as follows: Triticum monococcum. AA equal to n equal to 14. Azilopes, that is a wild goat grass, BB 2n is equal to 14. That hybrid is equal to AB. Hybrid results from capital A, capital B, that is hybrid is obtained AB 2n equal to 14, chromosome doubling, Amor wheat AA, BB 4n equal to 28, Amor wheat AA, BB 4n equal to 28, Agilops squarasa, a wild grass, that 2 DD 2n equal to 14 sterile, that is hybrid obtained ABD, that is 3n is equal to 21, Triticum aestivum 6n is equal to 42, it is a hexaploid modern wheat, A 6n Triticum aestivum, that is 6n is equal to 42, is a hexaploid modern wheat, which is obtained by chromosome doubling. Then another factor for obtained of here, that is polyploidy, is the origin of cotton. American cultivated species of cotton have 2n is equal to 52, and the Asiatic and wild American species cotton 2n equal to 26. It has been shown that American cultivated cottons were yellow tetraploids and arose as a result of crossing between two forms, followed by chromosome doubling in the hybrid. The long stapela, that is Narma cotton, Gossypium hirsutum, is an polyploid obtained by crossing Indian species, that is Gossypium herbaceum, with an American species Gossypium raimondi. Indian short staple, that is Gossypium herbaceum, and American short staple, that is Gossypium raimondi. Then AAA, that 2n equal to 26, DD 2n equal to 26. That it is the hybrid obtained AD, you have 2n equal to 26, chromosome doubling by colchicine treatment. That obtained is Gossypium hirsutum AAADD, that is Narma long staple, 4n equal to 52. Origin of tobacco. There is evidence that Nicotina tabacum is an allotetraploid between Nicotina silvestris and Nicotina tomentosa, or some allied species, that 2n equal 24. Nicotina tabacum is known only under cultivation and does not occur as a natural species. Origin of triticale. It is a man-made cereals and autopolyploidy triticum and sickles. So with this we finish this, that is polyploidy. Polyploidy means that origin that increase the number of chromosomal set and which results the size, leaf size larger, thick thickness of the leaves, and greenness is brighter, and fruit size is more large. Fruit size is more, pollen grain size is more. Every advantageous characters are due to the polyploidy. Every advantages. The productions is more, that is the result of polyploidy. If the size is more, then that is the advantages of polyploidy. So this is the advantage of polyploidy. With this we finish.